You are looking directly at an octopus. It is looking back at you. But here is what no one tells you. The arm reaching toward your hand right now is not following orders. No signal came from the brain. No decision was made at the center. That arm is deciding on its own. And the octopus has 8 more just like it. We have spent decades studying octopus intelligence. We have run the experiments, mapped the neurons, filmed the behavior. And the more we looked, the less the model made sense. Not because the octopus is mysterious in a vague, poetic way, but because the architecture of its mind violates something we assumed was fundamental. But intelligence requires a center, that a body needs one brain in charge. The octopus has approximately 500 million neurons, more than a dog, more than most birds. For an invertebrate, this is an extraordinary number. But the number is not the strange part. The strange part is where those neurons live. Only 1/3 are in the brain. The other 2/3 are distributed across 8 arms. Each arm contains its own dense cluster of nerve cells. Each cluster can receive information, process it, and generate a response, without consulting the brain at all. This is not a metaphor. This is the literal architecture of the animal in front of you. 140 million years ago, the ancestors of the modern octopus lost their shells. This is usually presented as a footnote, a transitional detail in an evolutionary timeline. But what actually happened in that moment was more significant than it first appears. The shell was not just protection. It was the reason the animal did not need to think quickly. With a hard exterior, survival was largely passive. You hid, you waited. The shell bought time. When the shell disappeared, time ran out. Suddenly, this soft-bodied animal, no armor, no skeleton, no rigid structure of any kind, was moving through an ocean filled with predators that could detect it, pursue it, and consume it in seconds. There was no margin. Every encounter with a threat required an immediate, local response. Not a response that traveled up to the brain, got processed, and traveled back down. That loop was too slow. A crab claw closing around a tentacle needed to be answered right there, in that tentacle, before the signal could even complete the journey to the center. This is the pressure that built the distributed nervous system, not as an experiment, not as an innovation, as the only available solution to a very specific problem. How does a boneless animal survive in open water when every second of delay is fatal? The answer was to stop centralizing, to push decision-making into the limbs themselves, to build a nervous system where the periphery could act without waiting for permission. The shell did not just disappear. It dissolved into neurons. Each of the octopus's 8 arms contains roughly 40 to 50 million neurons, arranged in a structure called a brachial nerve cord. This cord runs the length of the arm and connects to a series of ganglia, dense nodes of neural tissue that handle sensory processing and motor output locally. When an octopus arm touches something, the suckers respond first. Each sucker is lined with chemoreceptors and mechanoreceptors. They can taste. They can feel texture, temperature, pressure. This information does not travel to the brain. It is processed within the arm itself. The arm decides whether to grip tighter, release, or keep searching. In laboratories, researchers have observed something that takes a moment to absorb. When an octopus arm is surgically severed from the body, it continues to function. It recoils from painful stimuli. It reaches toward food. It attempts to bring what it finds back toward where the mouth used to be. For up to 1 hour. The arm is not running on residual electrical charge. It is not twitching randomly. It is executing purposeful behavior, approach, grasp, transport, in the complete absence of any connection to a central brain. The neural cluster inside the arm is still receiving input, still processing it, still generating output. This is not reflex. Reflex is a fixed response to a fixed stimulus. What the severed arm does is adaptive. It adjusts. It responds to what it finds. This is, by any reasonable definition, a form of thinking. The central brain, when it is connected, functions more like a conductor than a controller. It sets the general direction: hunt, hide, explore. The arms interpret that directive and execute it through their own local intelligence. When an octopus searches a rocky crevice for prey, it can simultaneously run multiple arms through different openings, each arm independently assessing what it encounters, each arm making its own decision about whether to advance or withdraw, while the brain attends to other things entirely. 9 parallel processes, running simultaneously, without conflict. Now consider this. The octopus is colorblind.
Its eyes contain only one type of photoreceptor. In humans, color vision depends on 3. The octopus has 1. By every standard model of vision, it should perceive the world in shades of gray. And yet it produces some of the most precise color matching ever observed in nature. Against sand, it becomes sand. Against coral, it becomes coral. Against a patch of algae-covered rock, it replicates not just the color, but the texture, the pattern, the specific spatial arrangement of light and shadow. For years, this was classified as one of the unresolved paradoxes of marine biology. An animal that cannot see color, producing color-accurate camouflage in under 1 second. The explanations proposed were complicated and unsatisfying. Then, in 2015, researchers discovered something that reframed the entire question. The same light-sensitive proteins found in the octopus's eyes, opsins, are also present throughout its skin. The skin itself is photosensitive. It does not form images the way an eye does, but it responds to light directly, locally, without routing the information through any central processing system. This is, again, not a metaphor. Skin samples removed from a living octopus and kept in controlled conditions will still cause chromatophores, the pigment cells, to change configuration in response to light exposure. The skin responds to light even when it has no connection to any nervous tissue whatsoever. This is a model that runs in direct opposition to vertebrate nervous system architecture. In vertebrate biology, seeing requires a dedicated organ connected by dedicated pathways to a centralized processing region. The signal travels in, the interpretation comes back out. There is always a center. In the octopus, the center is optional. The skin sees, the arm decides. The brain, when it participates at all, participates as one node among many, not as the origin of everything. If 9 semi-independent processing systems are running simultaneously, the immediate question is, why don't they conflict? When an octopus moves across a complex surface, reaching into crevices, wrapping around obstacles, extending and retracting different arms in different directions, there is no observable hesitation, no crossed signals, no moment where one arm pulls against another. The coordination appears seamless. But the brain is not producing that coordination through direct command. Neuroimaging and behavioral studies suggest that the brain issues what might be described as broad motivational states rather than specific motor instructions, something closer to intent than instruction. The arms receive that intent and resolve it into action through their own local computation. What prevents conflict, researchers believe, is partly structural. The neural architecture of the arms may include inhibitory pathways that prevent adjacent arms from issuing contradictory commands, and partly something that has not yet been fully characterized. The honest answer is that the exact mechanism of inter-arm coordination in the octopus remains an open question. This is not a gap that will be filled by minor refinement. It is a fundamental question about how a distributed intelligence maintains coherence without a central authority. And we do not yet have a complete answer. The leading framework for understanding the evolution of complex cognition is called the social intelligence hypothesis. In its simplest form, it proposes that large, sophisticated brains evolved primarily to manage the demands of social life, tracking relationships, navigating hierarchies, cooperating, competing, deceiving, and learning from others. The animals we typically identify as highly intelligent, primates, cetaceans, elephants, corvids, are all intensely social. Their cognitive complexity and their social complexity appear to scale together. The octopus does not fit this framework. For most of its life, the octopus is solitary. It does not maintain long-term social relationships. It does not live in groups with persistent hierarchies. In many encounters between octopuses, the interaction ends in predation. One eats the other. And yet, the octopus demonstrates cognitive abilities that place it alongside animals at the upper end of vertebrate intelligence. It navigates mazes. It solves multi-step puzzles. It remembers individual humans by face. It uses tools, not just single objects, but composite tools assembled from multiple components, a category that, until recently, was believed to be limited to humans, some primates, and a small number of bird species.
The ecological intelligence hypothesis offers an alternative explanation. It proposes that the demands of survival in a complex, unpredictable physical environment, finding food, avoiding predators, adapting to novel situations without the support of a social group, can drive the evolution of sophisticated cognition just as effectively as social pressure. The octopus, stripped of its shell and left to survive alone in a dangerous ocean, became intelligent because the alternative was extinction. This matters beyond the octopus. If intelligence can emerge independently along radically divergent evolutionary paths, once in the vertebrate line, once in the cephalopods, separated by something in the range of 500 to 600 million years of independent evolution, then intelligence is not a singular outcome that required a specific set of conditions. It is a solution, one that nature has arrived at more than once through completely different mechanisms toward apparently similar ends. In the autumn of 2019, researchers studying day octopuses on the Great Barrier Reef documented a series of interactions between a female octopus they had been tracking, designated Scarlet in their field notes, and a large coral trout. The trout is a predator. It is large enough to eat a small octopus. Under normal circumstances, proximity between these two animals would resolve quickly in one direction or another. What happened instead was recorded in detail. The trout approached Scarlet and changed its coloration from patterned to pale, a signal that marine biologists had previously observed in the context of cooperative hunting between different fish species. Scarlet observed the trout. The trout then oriented its body toward a section of reef and performed what the researchers described as a headstand, a posture used to indicate the precise location of hidden prey. Scarlet responded. She extended an arm into the indicated crevice. This behavior, using a directed gesture to indicate a specific object or location to another individual, is called referential signaling. It is considered one of the foundational components of symbolic communication. It requires the signaling animal to model the attentional state of the receiver. It requires the receiving animal to interpret the gesture as meaningful rather than incidental. The research team noted that Scarlet's response was not easily explained as a simple, conditioned reaction. The trout was not a familiar individual.
The signal was not one Scarlett had been trained to recognize. The behavior was context-dependent and appeared to involve genuine interpretation of the trout's intent. Dr. Alex Schnell, one of the researchers present, subsequently tested whether Scarlett would respond to pointing from a human observer. During a solo hunt, Dr. Schnell pointed toward crabs that Scarlett had not detected. Scarlett followed the gesture and caught the prey. The gap between the evolutionary lineages involved here is not a rhetorical flourish. It is a genuine measure of how differently these two nervous systems were constructed over how long a period in complete independence from one another. That two animals separated by that distance could share a communicative act, even a simple one, is something the existing frameworks for animal cognition do not fully account for. This response did not appear incidental, though its precise nature remains under investigation. In laboratory conditions, octopuses enter a sleep state. Their bodies become still. Their breathing slows. They are, by all behavioral indicators, unconscious. And then, something happens. Across intervals of what appears to be active sleep, a state that resembles REM sleep in vertebrates, their skin begins to change, rapidly. Waves of color move across the mantle. Chromatophores open and close in complex, shifting patterns. In some recorded cases, the patterns cycle through sequences that resemble the active camouflage displays the animal produces while hunting or hiding. If the arms retain a degree of processing independence even during waking activity, what happens to that independence when the central brain enters an unconscious state? Are the arms also sleeping? Are they running their own parallel processes? Is the color change during sleep generated by the central brain or by the distributed neural tissue in the skin and arms operating on their own while the center goes quiet? These questions are not rhetorical. They are open research problems. The neural mechanisms of sleep in cephalopods are poorly characterized. Whether octopuses experience anything during this state, whether there is something it is like to be an octopus moving through active sleep, is a question that the current tools of neuroscience cannot answer. We do not know what the 9 minds of an octopus do when the one in the middle goes dark.
Is the model of every other animal we have. It is the model our every other animal we have historically placed at the top of cognitive hierarchies. The octopus has run a different model for 500 million years. Not as a primitive alternative, not as an incomplete version of what we have, as a fully realized, extraordinarily effective architecture that has survived every mass extinction event in the fossil record, colonized virtually every marine environment on Earth, and produced, in complete isolation from the vertebrate line, a set of cognitive capabilities that we are still in the process of measuring. Control may not be a prerequisite for intelligence. We chose that model. The octopus chose differently. And the ocean, which has been running this experiment far longer than we have been asking the question, has not declared a winner. The arm is still reaching. And we are only beginning to understand what it means for a mind to have no single center.